Good afternoon. Welcome back. We are finally at day 3 of topic 19. This is probably one of the longest topics of the semester just because there's a lot of pieces and a lot of things to talk about, but we are finally almost done. So what we're going to do today is we are going to finish up part 2 and we're going to finish talking about DNA replication, specifically thinking about the replication machinery. So we're going to answer the question of how is DNA replicated? So before you all continue watching the lecture, one of the things that I encourage you to do is to go ahead and watch the video that is linked on this page. So for this video, this is an overview of the process of replication, and what it does is it talks about the main parts, the main machinery that is involved, and it gives you a really nice 3D overview of what is going on. So what I recommend doing is watching the video and then coming back and watching the remainder of this lecture, and then at the end of the lecture actually going back and rewatching the video just to help visualize some of these pieces in 3D. So what we're going to do is we're going to start with a practice problem. So for this practice problem, we are going to remind ourselves of how the heck do we figure out what is a 3' end and what is a 5' end of DNA. So this question is asking us to tell me which arrows are pointing to a 5' end. So if you recall back to our first part of our class, we talked about drawing ribose and deoxyribose. So for deoxyribose, it looks something like this. And recall that we start numbering from the carbon that has that CH2OH. So this becomes carbon 5, carbon 4, 3, 2, and 1. And so the 3' end is referring to the carbon that has an OH attached to it, and the 5' end, this, even though right now it has an OH, this ends up having a phosphate group attached there. And then recall to carbon number 1, this is where the base attaches to. And that 3' end is going to be really, really important today because as we will see, we can only add to the 3' end. So we talked about a quick and easy way to look to see where the 5' end versus the 3' end of DNA or RNA is to number your carbons really quick and look for where that phosphate group is. So if we take a look at D, we have a phosphate group there, and if we take a look at B, we also have a phosphate group there. So this is going to be a 5' end, and so is B. And oops, I wrote 3' instead of 5'. So both of these, the D and B, are our 5' ends, and because D is a 5' end, this tells us that A is a 3' end, and C is also a 3' end. So both D and B are correct for this practice problem. So before we get into DNA replication, there's a couple of rules per se that we are going to talk about that are going to help us through this process. And the first rule, if you will, is that DNA replication starts at something called an ORI, and ORI stands for origin of replication. So if you have a bacterial chromosome, which you're going to see me struggle drawing a double-stranded circular piece of DNA, let's make our ORI here. So here's our origin of replication. And so we're going to start replication there, and we're going to talk more about what this idea of ORI means here in a couple of pages. And the other thing that we will see is that as we showed in day 2 by studying Messelson and Stahl's experiment, that replication is semi-conservative. So we're going to see half old, half new. And we will also see that replication is bidirectional. So if we go back to our image here that we drew of our ORI, and let's zoom in on that DNA. So here's our double-stranded DNA, and let's go ahead and add our ORI on there, replication will move up and it will move down. So it will move in both directions. So that's what we mean by replication is bidirectional. When we read DNA, just like we read from left to right, there's going to be a way of reading DNA as well. And DNA is read in a 3' to 5' fashion. But, and this is a huge but here, when it is synthesized in a 5' to 3' fashion. And the reason for that is that polymerases can only add to the 3' end. And so this is why keeping our 5' and our 3' straight is going to be really, really helpful for us. So you will see me do my best to try to make sure that I label everything as 5' and 3'. Also, I'm going to apologize for the sound of my voice in this video. We spent about 6 hours on the phone yesterday with my side of the family and Garrett's side of the family talking to people. And even though I normally talk a lot, it was apparently a little too much for me. And then I have been trying to film a lot of lectures today just because last week was crazy busy. I was involved with doing a couple guest lectures for a directed study, and then I'm doing a couple guest lectures for that directed study this week as well. So it has been an insane couple of weeks, and I feel like even though I normally talk a lot, I've been talking a lot more than normal, and my voice is definitely starting to give out. So if I sound a little more quiet in the past couple of videos, and if you hear my voice kind of like sounding a little weird in this video, that is why. Apparently my voice box has been stretched to its limit. So now that we've got these rules, and you will hear me reiterate these rules quite a bit as we continue throughout this topic, let's go ahead and take a look at this origin of replication business. So we're going to start here on the left in this bracketed area, and this is for bacteria and archaea. So this is for our prokaryotic friends. And what's going to end up happening is we can tell right here we have opened up the DNA. So we open the DNA at the origin of replication and we begin replication. And we call this the replication bubble. And as we continue replicating, so as we continue going to the right, the dark DNA is new DNA, the light DNA is old. You'll notice that eventually here we get to the point where we have two new double-stranded DNAs. And so they're both half old, half new, so still following what we learned from Meselson and Stahl's experiment. But the reason that there is a difference between what is shown here on the left, and then let's change our colors here, versus here, what is on the right, here we have multiple replication bubbles. And here we've got a linear piece of DNA, and this is going to be in eukaryotes. And so the key here, and the reason that I'm showing you this, is that origins of replication exist in prokaryotes and eukaryotes. However, bacteria have one origin of replication and eukaryotes have many. And so in the case of the eukaryotic chromosome here, where replication has started and we've got a replication bubble here and a bubble here, that bubble, those two bubbles can eventually and will eventually continue growing and we'll get kind of a larger bubble. And so imagine a really long chromosome with multiple bubbles started everywhere. And a lot of this goes back to this idea that for bacteria, they have millions of base pairs and we have billions of base pairs. So by having more origins of replication, we can recruit more machinery and we can kind of replicate it a little bit faster, if you will. And then last but not least, I do want to talk about this electron micrograph that is here. And this is probably one of the coolest electron micrographs ever. But hopefully you can see these little arrows and where they're pointing. They are pointing to something known as the replication fork. And this might not mean much to you right now, and that is totally okay, because we will actually practice drawing replication forks and we'll talk more about them. But the reason this picture is so cool is because you can see the double-stranded DNA here, and this is the original chromosome. And then here you can see that it has been opened up at the origin of replication, and it has started to be replicated in both directions, and we can actually start to see that this will be a new strand, or that this will be a new chromosome, and this will be a new chromosome. So it's a pretty amazing picture. So with this knowledge, let's go ahead and do an application practice problem. So for the E. coli origin of replication, and really most origins of replication, they're going to be unusually rich in AT base pairs. In E. coli, it's about a 240 base pair DNA segment. So I want you all to go ahead and read the rest of the question and think about, why is our origin of replication mostly A's and T's? So hopefully you have had a chance to go through this question, and so we're going to walk through it together. So let's start with A. So AT base pairs can attract proteins better than GC base pairs. Well, AT base pairs and GC base pairs, they're really just purines base paired with a pyrimidine. We haven't talked about attracting proteins or anything like that. We can go ahead and cross that one off. So then we've got AT base pairs are harder to disrupt for B, and for C we have AT base pairs are easier to disrupt. Maybe, let's come back to those in a second. Let's go ahead and look at D. It's an accident of evolution. We can cross that off. And there is a reason why it is AT rich, and it has to do with disruption. So now let's see if we can figure out why it is easier to disrupt. Because if it was harder to disrupt than GC, well, if the origin of replication is where we open the DNA, that doesn't really make sense. So we can cross off B, and the answer is C, but let's think about why. So recall we talked about hydrogen bonds. We said that there are 2 hydrogen bonds between AT base pairs and 3 between GC. 2 hydrogen bonds is going to be a lot easier to pull apart than it is to pull apart 3 hydrogen bonds, which is why at origins of replications, we tend to have a lot of A's and T's. So hopefully I've convinced you that ORIs are very AT rich. So now we are ready for replication. So let's go ahead and define the idea of the replisome. Biologists love to add "ome" to every word. So we have things like genomics, we have proteomics, metabolomics. For whatever reason, adding that "ome" just makes that word feel much more intense and better. So the replisome, let's go ahead and define what the replisome is. The replisome is simply a complex of proteins that carries out replication. So pretty amazing process that happens with replication, as you hopefully saw in the little video clip, but the word is definitely much sexier than what actually happens, at least in my opinion. So for this process and for the replisome, just like many things, the enzymes are key. So we're going to have a lot of different enzymes that we're going to talk about. And I'm going to list the enzymes on this page, and then we'll actually talk about what they do over the course of the next few pages as we draw some of these pieces out. So parts of the replisome include an enzyme known as helicase. We'll also have single-stranded binding proteins, which are also shorthanded as SSBPs. We will have topoisomerase. We'll have a ligase, something known as the clamp or the clamp loader, and we'll have polymerases. So we'll have DNA Pol 3 and DNA Pol 1. So these are the 8 proteins that we're going to talk about. Most of them are enzymes, and we will talk about their functions. So what we're going to do is we're going to do two things. We're going to draw a picture of the entire replisome, and after we draw a picture of the entire replisome, then we're going to start on a strand of DNA, and we're actually going to go through step by step what happens. Okay, so let's go ahead and start with a brand new blank page. And on this page, this is the page that we are going to draw the entire replisome on. So we're going to draw an open strand of DNA. All right, so now that we have this open strand of DNA drawn, let's go ahead and throw some labels onto here. So we're going to make this top strand 5' and this bottom strand 3'. So we have drawn this strand of DNA open. And the reason that this strand of DNA is open is because we had a protein called helicase, and helicase, its entire job is to open the DNA. And what it does is it breaks those hydrogen bonds that hold together the A's, the T's, and the C's and G's. So if this was an origin of replication, we would expect there to be a lot of A's and T's. So now that we've got the DNA open, well, these bases are complementary. They want to stick close and they want to come back together. So what we have are these little proteins, and I'm going to draw them like beads on the outside here. And these are the single-stranded binding proteins. And the single-stranded binding proteins, their entire job is to keep the DNA from resealing. Because otherwise, what would be the point of having our helicase come and open the DNA? Now we're going to have another protein that is probably somewhere around here. You'll notice here I kind of drew this tangle in the DNA. You can imagine if you're taking DNA that is a helix and you're opening it up, the more that you open it, the more tangled it gets, right? So back in the day when we all had headphones that still had cords onto them, before Apple took away our aux input that we had on our phone, so our 3mm plug that we had, before they took that away, when you lost your headphones in your backpack or your purse and you went to grab them, if you try to pull them apart, they would just get more tangled, right? It was a huge pain in the butt. Well, the same thing happens with DNA. When we open the DNA up and we start pulling it open, we start to get tangles. So we're going to have a protein that is called topoisomerase, and its entire job is to relieve the supercoils or to untangle DNA. So it plays a pretty important role. All right. So now that we've got our DNA open, helicase is continuing to open it, topoisomerase is going to continue to relieve any of the supercoils or untangle DNA, we're going to start adding, we're going to start adding a couple more pieces. So let's go ahead and we're going to make pink our primer, and specifically we lay RNA primers, and then we'll do bright green. Can we see that? Yeah, okay, perfect. We'll do bright green as new DNA. So we're going to lay a primer. Now, the protein that lays a primer is called primase. So I'm going to add a little primase protein here. And recall that when we make DNA, or when we make any sort of nucleic acid, even if it's not DNA, we are going to read DNA from 3 to 5, and we can only synthesize nucleic acids from 5 to 3. So here we have a 3' end, which means our primer has to start at 5', and we're going to lay a short little RNA primer. So here's our RNA primer, and this RNA primer is laid by primase. And primase, just like our polymerases that we're going to talk about, can only add to the 3' end. And it's just a function of these polymerases and how they work. So now that we have an RNA primer, now we are primed and ready for our DNA polymerase. So let's go ahead and add some primers on our top strand. Now, our top strand, we have a little bit of an issue here. We have a 5' end here, which means somewhere down here, if we were to follow this along all the way to the end, this is our 3' end. So that means when we have new DNA that is laid right here, this right here must be the 3' end. So if we can lay primers and we can only lay them in the 5' to 3' direction, that means we are going to have to lay our primer further up. So here is our other primer that we laid, and now both of these strands are primed and ready to go. So let's go ahead and draw our polymerase. So we're going to draw, it's kind of got 2 arms here. So here is DNA polymerase 3, and this is our main polymerase. This completes the synthesis of DNA. So this is going to read the DNA in a 3' to 5' fashion. It finds this free 3' end, and it's going to synthesize DNA all the way to the end, and there's our 3' end there. On this bottom strand, it'll start here at that primer, and it'll continue synthesizing into the fork, and this will be our 3' end. Now there's another protein that its job is to help the polymerase stay on the strand of DNA. So we're going to draw kind of a little half circle here, and this is the clamp or the clamp loader. And again, all this does is it helps keep the DNA polymerase on the strands. So helps from slipping, if you will. So now that we've got this, we're going to go ahead and this bottom strand that we've drawn down here, we're going to call this the leading strand. And we're not going to define what that means yet. We're going to wait a little bit, but I want this picture to be all inclusive. And we're going to define this top strand as the lagging strand. And again, we will come back to this and we'll talk about why this is called that. So let's now return back to our primers here. For our primers, those are RNA. We need our new DNA to be all DNA. We don't want a mix of DNA and RNA. So we have another polymerase. So let's draw another little colorful protein here. This is going to be DNA Pol 1. And DNA Pol 1 will remove the RNA primers and it will lay DNA. So it'll fill it in with DNA. Well, when it does that, we end up with a little bit of a problem. So let's go ahead and take this 3' end here where there used to be an RNA primer, and let's zoom in on that for a little bit. So let's go ahead and draw our DNA. And all right. So what we have is we have this gap here. This piece right here used to be primer. This piece was where DNA polymerase 3 started. DNA Pol 1 came in right here, removed the primer, and filled in DNA. Well, now here, there's a nick. And this nick is in our phosphodiester backbone. We don't want a nick. We want a continuous phosphodiester backbone. We want all of those covalent bonds to be formed. So we have one more protein, and this protein is called ligase, and ligase is oftentimes referred to as the molecular glue, and this is going to fix the nicks. So anywhere where there is a nick because there was a primer and we had to remove it and then put in fresh DNA, that is going to be fixed by ligase. All right, so this image, although great because it has all of the pieces on it, it's a little hard to look at. It's a little hard to understand everything that's going on. So we're going to simplify this. We're going to take a step-by-step approach now. So let's go ahead and draw a piece of DNA opened up. And the first thing that is going to happen is we will have helicase come in. So our helicase is going to open the strands of DNA at the origin. And then, let's actually be consistent. So let's use green. And then we'll have our single-stranded binding proteins come in, and they're going to keep the DNA open. Now that we have our DNA open, kind of step one, if you will. Now that we have the DNA open, now we are ready for primase, and primase will lay an RNA primer. Now that we have laid the RNA primer, now we are ready for the clamp protein and for DNA polymerase. So we're going to draw kind of a tangled piece of DNA here because we've started opening things up more and more. You can probably tell what protein's going to come in here next. Okay, so we're going to have a couple of proteins down here. We're going to have our topoisomerase, and our topoisomerase is going to untangle the DNA. We are going to then have our polymerase. So DNA Pol 3 uses those RNA primers to start DNA synthesis. And then the clamp loads the DNA polymerase. So this clamp will load the DNA polymerase, and then DNA polymerase is going to use the RNA primers to start DNA replication. So what we're going to do now is we're going to zoom in on the bottom strand. So we're going to forget about the top strand for now. We will come back to it, but for now, let's just focus on the bottom strand. So on a new page, I'm going to go ahead and draw what our bottom strand looks like so far. So our old DNA, we had that drawn in a black color, and we had our primer drawn in pink, and we'll be consistent with our giant drawing that we did, and we'll make bright green our new DNA. So our DNA polymerase 3 came in, it used that primer, and it kept going, and it synthesized new DNA. So now we have half old and we have half new plus a primer, right? We have that little RNA primer. Well, now we're ready to get rid of that RNA primer. So we are going to have DNA Pol 1 come in, and what DNA Pol 1 does is it's going to remove those RNA primers and replace with DNA nucleotides. All right, so hopefully you have caught up to where I am. So now what we have after DNA polymerase 1 came in and removed that RNA primer, well, now we have that nick. So we have a break in the phosphodiester backbone. And so we have yet another enzyme that comes in. And the enzyme that comes in is ligase. And ligase is the molecular glue, and it is going to fix that gap or nick in the phosphodiester backbone. So then at the very end, I'm going to kind of shorthand this here. What we are left with is a full half old, half new DNA strand, or I should say double-stranded DNA. So we've got half old and half new double-stranded DNA that we are left with. So we have been focused on one of the strands, but if we go back to either this image or the first one that we drew, here we have a replication fork. And we mentioned this idea of a replication fork even when we first started looking at our origins of replication. So here are our replication forks. So when we open a replication bubble, or when a replication bubble is formed, we have two replication forks for every single bubble. Well, we have only been focused on one replication fork, so we've only been drawing one of them, and we've only really focused on one strand. So let's go back for a second and let's actually draw a true replication fork, and let's draw what actually happens, and this will take us back to this idea of the leading strand and the lagging strand. So we're going to start with drawing double-stranded DNA, and it will be open into a replication fork. Keep in mind, we're only looking at one. Theoretically, right? We have another one here because this is our replication bubble, but we're just going to focus on 1/2 for now. So when we have this 5', 3' end, this has been opened by helicase. So let's go ahead and just draw helicase in here just to remind ourselves that it's going to continue unzipping the DNA. We're going to kind of simplify the rest of it. Otherwise, we would be here for eons drawing the entire replisome each time. So let's just keep it pretty simple. We're going to keep pink as our primer, and we will keep green as our new DNA. So we're going to lay a primer, and again, because of our rules of reading 3 to 5 and synthesizing 5 to 3, we have an RNA primer that is laid from a 5 to 3 direction, and we'll have an RNA primer that is laid in a 5 to 3' direction on the top strand as well. So now when DNA polymerase comes in, it's going to start, so let's start with the top strand. It's going to start here at this 3' end, and it's going to replicate away from the fork. So our fork is right here where helicase is. On the bottom strand, our 3' end is right here, and so we are going to replicate into the fork. All right, so we have drawn a snapshot of replication. Now, let's go ahead and open the DNA more, because recall, helicase, its job is to constantly be unzipping that DNA and opening it up. So by the time we lay the primers and we start synthesizing, more DNA has opened up. So let's go ahead and open the DNA even more. So this is still the same replication fork. It is just larger. So here's our fork. It has now moved to the left because our fork is moving this way. So let's get our original primers on here, and then let's get the DNA that was already synthesized on here. Okay, so let's start with the bottom strand. So for DNA polymerase, we said that DNA polymerase can only add to the 3' end. We have a 3' end here, so check, meets that description. And DNA polymerase can only add where there is double-stranded nucleic acid. So we have double-stranded nucleic acid there, right? We have DNA here and we have DNA here. And so check, DNA polymerase has everything that it needs. So on this bottom strand, we are going to continue replication still into the fork. We do not need anything special as that replication fork opens, because we have a 3' end and because we had double-stranded nucleic acid. Up here, we had a 3' end and double-stranded nucleic acid. Granted, it was a hybrid because we had DNA and RNA, but that's okay. DNA polymerase doesn't care. That's totally fine for it. It was able to go ahead and start replication here. So bottom strand's good. It's got that 3' end here, and it can replicate into the fork. Because it does what we call continuous replication, and the reason it can do this is because it requires one RNA primer, and you always have a 3' end. We call this the leading strand. And so the reason it's called the leading strand is because it's leading DNA in the sense that it's going faster than the top strand. So speaking of our top strand, we've kind of ignored it. Let's go back to it and let's take a look at it. So we've got double-stranded nucleic acid here, right? We've got DNA and RNA. So that's a check for polymerase. But wait, we have a 5' end here. Well, crap. Can we add to a 5' end? Heck no, right? DNA polymerase needs that free 3' end to be able to add. So, well, we've got a problem. Well, we have a problem, but we have all of this opened DNA here. And we have a special enzyme that can prime things for DNA polymerase. So what if we come in and we lay a primer here? So here's our 5' to 3'. And the reason I know this is a 3' end is because I have a 3' end right here, and DNA is anti-parallel. So now DNA polymerase, it's got double-stranded nucleic acid, check. Free 3' end, check. So now we can fill in this piece of DNA with DNA polymerase. So what we have here is we have these fragments, and these fragments are of DNA and RNA primers, and they actually have a special name. They are called Okazaki fragments, and they are named after the scientists that discovered them. And because this strand has to have a primer laid, and then we fill in the DNA gets opened more, we lay a primer, fill in the DNA. You can imagine this takes a long time. This is called the lagging strand. And the reason it's called the lagging strand is because it lags behind that previous strand or the leading strand that is able to do continuous replication. So we say that we have discontinuous replication on the lagging strand. So this idea of a replication fork there's a lot to it. So we're going to do a lot of practice problems. So let's go ahead and take a look at a textbook picture here. And what this does is it again shows us and walks us through the 5' and 3' ends. So let's go ahead and start at the bottom here. So this red is supposed to be our primer. So here's our 5' end, which means right here must be a 3' end because DNA is anti-parallel. Up here we have a 3' end, which means this must be a 5' end, and our top strand is lagging because every time that DNA fork opens up more, we have to lay a new primer, which is shown here, and then we have to fill in. So our top strand is lagging and our bottom strand is leading. Now what I encourage you to do is I encourage you to draw these. So before we go on to drawing these, we're going to fill out one more practice problem together. And on this one, nothing is labeled. So we have to figure out all of the pieces. And in addition, this one's also flipped the other way. So the best way to test yourself with replication forks is to draw them in all sorts of different directions. And if you can draw it up, down, left, right, northeast, northwest, then that means you definitely have it down. So let's go ahead and why don't we start with our top strand right here. So this red is our primer, and the arrow indicates the direction of synthesis, and we know we can only add to the 3' end. So this makes this end the 3' end, which makes this end the 5' end. We then know because DNA is anti-parallel, because this is a 5' end, this right here must be a 3' end. Because this end is a 3' end, this blue strand and this blue strand were originally paired. Therefore, this is a 5' end. If you don't believe me that way, we can also actually head over here to this primer. The arrow points the directionality of synthesis. So that's the 3' end, which means this is the 5' end. And because this is the 3' end, this right here is the 5' end. So different ways you can figure out what's the 5' and what's the 3' end. So we have our 5' and 3' ends labeled. Now let's figure out which one is the leading and which one is the lagging strand. So when we come in and we make new DNA, we're going to be able to make new DNA in this direction. So here's our new DNA. We are going into the fork. Anytime you are moving into the fork, you are the leading strand. On the bottom strand, we are going away from the fork, and this is our lagging strand. And if you don't believe me, we can actually go ahead and, what happens if we open this DNA up even more? So if this DNA is opened up even more, we're going to draw kind of an awkwardly shaped fork here. So here's our fork. Here was, let's make sure we match our colors. Here was our original 5' to 3' primer, we had DNA that we added, and then on the bottom we had an RNA primer going 5' to 3', and we added DNA. Well, now that we're open, we can go through our checklist again. So DNA polymerase is going to hang out here. Free 3' end, check. Double-stranded nucleic acid, check. It meets all of its requirements. It will continue replicating. So we have continuous replication. We do not need another primer. Let's take a look at our bottom strand. Do we have double-stranded nucleic acid? Yes. Do we have a 3' end? No. So we cannot continue there. So what we're going to do is we're going to bring primase back. Primase will lay another primer for us. And then we'll backfill and fill this in, and this is where our Okazaki fragments will come in. So what I'm going to do on these 2 pages here is I am going to draw 2 forks, and I'm actually going to draw them both the same way. So they'll both be facing up, and I will only label one of the ends. I should be able to give you a replication fork with only one strand labeled, and you should be able to fill in the other strands and tell me which one is the leading and which one is the lagging. So when I post these notes, I will actually write out the answer to these practice problems, but I encourage you and I challenge you to try to walk through these on your own. All right, so we're almost done with this idea of copying DNA. But one of the questions that we have to answer and think about is, well, how the heck does the polymerase know which nucleotide to add? Well, we've already answered this question. We have said that the bases are complementary. And so we have said that the polymerase is going to read DNA 3' to 5', and it's going to make DNA 5' to 3'. So let's write an example. So let's do 3' to 5'. Let's do AT, A, C. So this will be our old DNA. Because we know what the old strand is, we can figure out what the new strand will be. We're going to have 5' to 3' because DNA is anti-parallel and complementary. A pairs with T, T pairs with A pairs with T, A pairs with T, and C pairs with G. So this is going to be our new strand. So because the bases are complementary, the polymerase can read DNA and it knows what nucleotides to bring in. So let's do a quick practice problem. So here is a strand below, and we're going to figure out what the complementary strand of DNA would be. And sometimes people say complement the following strand of DNA, figure out what complementary strand or complement the strand. Those are both used interchangeably. So we know DNA is anti-parallel, so we can put our 5' and 3'. So now we're going to go ahead and complement this. So we've got A pairs with T, G with C, C with G, T with A with T, A, C, G, A, T, G, C. So we started with a 3', so we can actually cross off A and B. Those don't even have the right end labeled. So that leaves us with C, D, and E. And A pairs with T, so we can actually go ahead and cross off C and D, which leaves us with E. And if we double check E to make sure that it matches with what we complemented, it in fact does. And so the complement of this black strand is the purple strand, which is E. All right, so a couple more things before we finish up. Let's talk about the action of the polymerase. So this is really important. This is going to be a throwback. So you probably thought we were done with reaction coordinate diagrams for the semester, but not so quickly. So let's take a look at what's going on here on the right. All right, so we are reading 3 to 5. Synthesizing 5 to 3. So here we've got our 3' end that we're going to add to. Here we have our A, And here's our next nucleotide, a T, and you can actually start to see the hydrogen bonds here. And you'll notice that we have 3 phosphate groups here, but a nucleotide has 1. So in order to understand the action of the polymerase, let's think about how nucleotides are like ATP. So let's recall what happens to ATP. Well, ATP is adenosine triphosphate. So if you recall from our studies of ATP, when we hydrolyze ATP, we go to adenosine diphosphate plus a phosphate group. And here we have a delta G that is negative. So this is an exergonic reaction, and this is favorable. So how can we take this and how can we apply this to what happens with the polymerase? So what the polymerase does is it's going to use DNTPs. And DNTPs are deoxyribonucleotide triphosphates. And we're going to add to the 3' end. And if we peek at this picture, this actually gives us a hint here. We have 2 phosphates that are released. So we release 2 phosphates. So for a DNTP, we're going to have a sugar with a base, and we have 3, I ran out of room. Here we go. We've got 3 phosphates that are attached to that. So if we draw a reaction coordinate diagram, we will have a, let's say, DATP. So we will have deoxyadenine triphosphate, and we will get deoxyadenine monophosphate plus 2 phosphates that are released. And so here is our delta G. It is less than 0, which means delta G is negative. And so the way that the polymerase works is by use of the DNTPs and release of 2 phosphate groups. This is going to release energy, and that's going to make delta G negative, which is going to drive the reaction forward. And so the DNA polymerase, if we think back to our other little piece here, our activation energy, the DNA polymerase is going to lower the activation energy, and it's going to align the bases. So by aligning those bases, it makes sure that they are complementary, and it helps to lower the activation energy, and that energy that is in the bonds of those DNTPs is used to drive this reaction forward, because remember that building nucleic acid is an endergonic reaction. So just like everything else in this class, an exergonic reaction has to be coupled to an endergonic reaction. So we use the energy that is stored in those DNTPs to drive this reaction forward. Okay, so we kind of have an issue here, right? Let's recall and remind ourselves of what the difference between eukaryotic and prokaryotic chromosomes is. All right, so hopefully you have recalled that a prokaryotic chromosome is circular, whereas a eukaryotic chromosome is linear. So do you see a problem yet? If not, that's okay. Let's go ahead and see if this practice problem helps jog your mind a little bit. So here on the top, what we have is we have 2 strands of DNA. So we'll go ahead and draw this out. So here's our old DNA, and we'll keep our colors consistent to the ones that we've been using. Pink will be our primer, and then green will be our new DNA. So we've got 2 old strands, and we're just going to draw what they look like at the very end. Okay, so after all of our replication has gone through, this is what our strands look like. So we've got 2 primers left, right? We don't want RNA primers. So we kind of have an issue. What happens if we remove this RNA primer? Isn't this end right here a 5' end? Do we have a way to fill this with DNA? What about this one down here? If we remove this pink, well, this end of this green right here, this is a 5' end. Can we add on to a 5' end to backfill this? No, right? We call this the end problem. So for eukaryotic chromosomes, they're linear. We have this end problem. So based on the figure, both the picture from the textbook on the left and the figure that we drew together on the right, we have a problem. We can cross off A, nothing, the chromosomes are replicated without any problems. Well, that's false. We just said we have something called the end problem. B, the chromosomes become longer. How can they become longer if we're missing and we're not replicating a chunk of a chromosome? So that's false. So the answer is C. The chromosomes become shorter in each round of replication. When we remove this primer, we are left with this 3' overhang of old DNA, and we have absolutely no way to replicate it because DNA polymerase cannot add to the 5' end. So our chromosomes are going to become shorter with every round of replication. Now you can imagine that we don't want that to happen, right? We have really important information in our chromosomes. So luckily for us, there is something called telomeres and telomerase. So here on this picture on the right, this is just showing us the end problem again. So here is this gap that we have, and we have another gap here. So again, this is just this idea of the end problem because we cannot go back in there and we can't prime the ends and fix it. So we have two things. We have something known as telomeres, and telomeres are specifically the ends of our chromosomes. And we say that they're protective ends. And they're pretty cool because they are a series of non-coding G-rich sequences. So what we have are, we're going to call this the important stuff. So all of our important genes. And then we have a crap ton of G's. So, which means, of course, our complementary strand is going to have a lot of C's on it. So we have these non-coding G's. And so those are the telomeres. And again, they're just there to serve as a protective mechanism. Another player that we have is we have telomerase. So ends in ase. This is an enzyme. And so what this enzyme does is this enzyme fixes the end problem. We're not going to get into the specific mechanism of the enzyme. It's a little outside of the scope of this class, but the way that it does it essentially is it's going to fill in the gaps and it helps maintain the telomere length. So our chromosomes do end up still getting a little shorter. So if you were to look at the chromosomes of an infant compared to when they're older, they'll be a little bit shorter. This isn't perfect, but it is good enough. And so this enzyme is going to fix these gaps that we have and help solve this problem of us losing important parts of our chromosome. So believe it or not, we have finally survived and made it to the end. So what I encourage you to do is, on random scratch sheets of paper, is go ahead and just draw the process of replication. If you're eating your breakfast and you are finding yourself looking for something to do, go ahead and doodle replication forks and make sure that you can label those 5' and those 3' ends. So congratulations, you have survived the history of DNA and DNA replication. I know that it is a lot, so I encourage you, if you need help, please don't hesitate to reach out. I am more than happy to help in any way that I can. So thanks for hanging out with me, everyone, and I will catch you in the next topic. Have a great day.